Welcome back, students. In this video, I am going to talk about vegetative propagation. So, in multicellular organisms, that is in plants, asexual mode of reproduction exists. It means from a single parent, without any gamete formation and without any fertilization, new individual plants can be formed. So, how they can be formed, let us discuss in detail now. So, the part of a plant which is used here, it is called as explant. So we usually call it as explant. Okay. So from this part of a plant, we are going to produce a new individual plant under controlled conditions. It means we grow this part of a plant under special media. It means we provide some nutrient supply to it, that we call it as special media, as well as we maintain temperature, okay, as well as we grow usually them in the microbe free environment. Microbe free environment. Okay. So the part of a plant from which the new individual plant is developed and under controlled conditions is known as vegetative propagation. And the ability of the plant, we call it as totipotency. The ability of a plant to generate a whole plant, that is called as totipotency. Okay. The ability of a plant part or cell to generate the whole plant is called as totipotency. And this process is called as vegetative propagation. So what are the advantages of vegetative propagation? All the plants are genetically similar. And secondly, they bear fruits and flowers early than the plant that has grown from the seed. Suppose if the plant, this part of a plant age is 5 years, the new plant which is produced, this age starts from 5. But if you take a seed and grow it, it starts from the day 0. So that is the difference. So because of this difference, these plants which have grown by vegetative propagation, they bear flowers and fruits very fastly. And propagation of seedless plants. Without any seeds, we are developing the plant. That is called as propagation of seedless plants. So if there are advantages, there will be some disadvantages also. So what are they? It means there is no variations. All plants are similar. It means there is no variations. So these plants will be more sensitive. Why they are more sensitive? Because they are grown under special conditions. As they are grown under special conditions, these plants will be more sensitive in nature compared to the plant which is grown naturally. So you can see this example of bryophyllum. So this is the leaf of bryophyllum. The tip of the leaf contains some buds. The tip of the leaves contains some buds. So when these buds fall onto the soil, they grow into an individual plant. When these buds fall onto the soil, they grow into an individual plants. So this is one of the best example for vegetative propagation. In higher classes, we will be studying more examples. So now let us talk about one more concept under vegetative propagation, that is tissue culture. So it is a laboratory technique in which part of the plant is grown on the artificial medium containing hormones under controlled conditions. It means in a test tube, we have the media which is containing required nutrients as well as hormones. Okay, so we will put the part of the plant here, and under controlled conditions, this will be grown into a whole plant. So this process is called tissue culture. So it has different steps. First, we need to collect the sample from the parent plant, and then we put them on the agar plate, and this agar plate contains nutrients and auxins. Auxin is a plant hormone, right? So when it is supplied with the nutrients and the plant hormones, it starts growing into the plantlets. So small leaves will be developed. So when the small leaves are developed, so these plantlets are transferred to the soil, which is containing the compost, or again it is supplied with the required nutrients as well as hormones. So this is called as tissue culture. Next, let us talk about the one more technique of vegetative propagation for developing varieties. It is grafting. So you can see here, this is a type of vegetative propagation in which stems of two plants are exchanged and joined together. It means we take two different plants, and cut them. So this is plant 1, this is the stem of plant 1, and this is the stem of plant 2. Okay. So what we'll do, let us assume this is the stem, both are the stems of mango plant. This mango plant is sweet in nature. These mangoes are sweet in nature. These mangoes are in very good size. Okay. These mangoes are large in size. Okay. So as this mango, so we want the plant which is giving large size plus sweet mangoes. So what the farmer has done, so he has cut the stem of this plant 1 as well as plant 2, and he has inserted this and tied it. Okay, so the plant which is in the upper region, this is called scion, and the plant which is in the lower part, that is called stalk. Okay, so we need to make sure that the xylem and phloem are continuous. So what we need to make sure, xylem and phloem are continuous. So only if the xylem and phloem are continuous, this will be survived. The upper scion part will survive. Otherwise, it will get dried and it will die. So the upper part is called scion and the lower part is called stalk. So the plants which are generated in this way and the mangoes of this plant will have both the characters, that is large size and sweet. So this way, without any scientific knowledge, new hybrid varieties can be developed. Okay. So if you write any entrance exam, they may ask, write the type of, which type of asexual mode of reproduction is employed for producing new varieties. So that is grafting. So now let us talk about spore formation. So spores, usually under unfavorable conditions, any organism which is forming spores, it will form under unfavorable conditions. What are unfavorable conditions? When the environment is not good for its growth and development, or when the nutrients required are not available, in those cases, these organisms will develop spores. Okay, spores are the structures which can survive under unfavorable conditions. And when they come in contact with the favorable conditions, or that means when they come in contact with the nutrients or the suitable environment, they grow into a new organism. So now let us see in detail. So under unfavorable conditions, cells break up into tiny structures called spores. So these spores, why they are survived in unfavorable conditions? Because they have thick walls that protect them. So as in case of plasmodium, you can see there is one cyst, and inside this the nuclear divisions were going on, right? So in the same way, these spores are also covered by a thick wall which protects them in the unfavorable conditions. When these come in contact with the favorable conditions, they grow into an individual plant. Example is rhizopus. So coming to the questions, what is vegetative propagation? States its advantages as well as disadvantages. Describe the reproduction by spores in rhizopus. Thank you for watching the video. In the next video, I will introduce you to the sexual reproduction